Hey, 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 let's talk about the cell cycle. All right, so if you're a cell and you are unicellular, when you reproduce, you are reproducing the whole organism. However, when you're multicellular and you reproduce, if you're a multicellular organism and you reproduce some cells, it could either be for growth of that whole multicellular organism or repair for that whole multicellular organism. Now, in order for that multicellular organism to reproduce, uh, organismally reproduce, what they're going to need to do is undergo meiosis and then cell division in order to create gametes, which would then be involved in sexual reproduction if you're a multicellular organism. So, if you're a prokaryote, that means that you do not have a nucleus. And so their process of cell replication is actually much more simple than eukaryotes. So they undergo this process called binary fission. So the first step, the DNA will replicate, and typically it's going to just be one circular chromosome, one large circular chromosome that composes the genome for that prokaryotic cell. What happens after that is that DNA, that replicated DNA, will then attach to the plasma membrane on either side, and then cytokinesis will occur, splitting that cytoplasm down the middle. However, if you're a eukaryotic cell, well, there's a few ways you can go about it, or go about cell replication. There's two different reasons why you would. So if you're a somatic cell, for example, you're first going to undergo a process called mitosis. So mitosis, what that's going to do is that's going to result in two genetically identical nuclei, which will then eventually lead to the division of the cells. So two, hopefully, genetically identical daughter cells. Now, if you are trying to undergo, some kind of sexual reproduction, you're going to use a process called meiosis. Meiosis is going to result in four genetically different nuclei, which then leads to four genetically different cells with only half of the genetic material, with half of the DNA. We would call those types of cells gametes, and because they only have half the DNA, they're considered to be haploid, or 1N. So again, this is eukaryotes, and because eukaryotes are a little bit larger, a little bit more complex, and importantly have nuclei, they're going to have these two processes, mitosis or meiosis, depending on what you're trying to divide. So mitosis and meiosis only refer to the division of the nucleus, as a fun fact. All right, so just overviewing the cell cycle. So the cell cycle is going to encompass all of these terms here. Your interphase, part of the cell cycle, mitosis, part of the cell cycle, cytokinesis, still part of the cell cycle. Now within interphase, to break down interphase a little bit, during interphase, all of your genetic material is going to be in chromatin form. So that's going to be this haphazard kind of spaghetti-like form of your DNA with a lot of surface area. And with all that surface area, it exposed, it's going to be easily picked up by enzymes or proteins involved in gene expression. So in fact, during interphase, you're actually going to have a pretty high metabolism, and specifically during your G1 phase of interphase. So interphase is composed of three subphases. During G1, your cell will be undergoing cellular growth and conducting its normal cellular processes, so it's going to have pretty high metabolism. Now, during G1, if you receive some kind of, for example, exogenous signal to replicate, you're going to transition from G1 to S phase. So I like to associate the S and S phase with synthesis because you're going to be synthesizing new DNA. So S phase, what happens is your DNA is going to replicate. So you're going to duplicate all of your DNA in anticipation of eventually reaching mitotic phase. Now, after your S phase, you're going to transition into G2 phase. And your G2 phase, what's going to happen is you're going to grow a little bit more in preparation for mitosis. And then mitotic phase, that's actually the whole process, multi-step process of nuclear division. And then lastly, after your nucleus has divided, you're going to undergo a process called cytokinesis, which is actually the division, the physical division of the cell. All right, so speaking of moving from phase to phase, let's talk about how this happens mechanically. So a protein complex containing cyclin and cyclin-dependent kinase, or cyclin CDK, transitions the cell from phase to phase. All right, so cyclin, that's actually just going to be a type of protein. And if you notice here, you have cyclin D, you have cyclin E, you have cyclin A, you have cyclin B. So these are just proteins. Now these proteins, what they do is they have this capacity to activate these things called cyclin-dependent kinases, or CDKs. So cyclin-dependent kinases. Kinase ends in an ase, so we know it's an enzyme. What this enzyme does is it phosphorylates things. So by phosphorylating something, what you're doing is you're adding a phosphate to something. It could be a protein. And typically, what adding that phosphate to that protein does is it either activates or deactivates that protein. All right. So these cyclins are simply activators of your kinases. They're activators of these enzymes. So these enzymes are normally floating around in your cell, but they only become active when they meet their counterpart. So cyclin D, if you notice here, we have cyclin D, which is bound to CDK, or cyclin-dependent kinase 4. Cyclin E is bound to CDK2. Cyclin A is bound to CDK2. Cyclin B is bound to CDK1. We don't need to necessarily know the numbers associated with the CDKs. What we do need to know is that you have different cyclins, and those cyclins will have different counterparts that they activate. Okay, so during G1 phase, you're producing cyclin D. So cyclin D, remember, that's a protein that activates a kinase. So cyclin D is produced. That cyclin D will bind to its counterpart, so in this case CDK4. So it's going to bind to a CDK, a cyclin-dependent kinase. And what happens is when that cyclin-dependent kinase is activated, it's going to lead to a signal transduction pathway. And what a signal transduction pathway, in short, is pretty much, you're a protein and you bump into another protein in a way that might change its conformation. And by changing its conformation, I just mean changing its three-dimensional shape. It could activate it, it could deactivate it. In this case, we'll just say it's activating another protein that it bumps into. Then that protein that it bumped into, well, because it was bumped into by the previous protein, has changed conformation and now has an affinity to bump into a different protein. And that other protein will receive that bumpage of the previous protein, and that might change its conformation, allowing it to more efficiently bump into another protein, changing its conformation, and so on and so on and so on and so on, eventually until you get gene expression, a change in gene expression, from the nucleus. And so what happens, so going back to it, cyclin D is produced during G1, which is bound to its kinase counterpart, its CDK. Okay, so you have this cyclin D CDK complex, which then initiates a signal transduction pathway that leads to the expression of cyclin E. So eventually, protein bumps into protein, bumps into protein, bumps into protein, and eventually you get the expression of cyclin E. So cyclin E, that's a protein that's activating a cyclin-dependent kinase, or CDK. So cyclin E is going to bind to its counterpart and activate its counterpart, activate its CDK. And that's going to initiate a signal transduction pathway in order to express cyclin A. So cyclin A is another protein that will then activate its CDK, bind to its CDK, creating that complex, initiating a signal transduction pathway that would eventually lead to the expression of cyclin B. So that's kind of how you transition from one phase to another. Fun fact, your cyclin A is actually involved in expressing all of the proteins associated with DNA replication, which is actually precisely what happens in S phase. So here I have labeled a restriction point. There's a few restriction points within the cell cycle, but the one that we're going to talk about on the next slide is this transition between G1 and S phase. So restriction point is just a point where you have cellular checks, you could say, or the cell is inhibited chemically from transitioning from one phase to another. And until all of the criteria are met in order to transition from one phase to another, it will remain in its original phase. So it remains in G1 until told otherwise. So we're going to go under a few of the things that would affect the transition between G1 and S. So just as a review, CDK, that's a cyclin-dependent kinase. So it's a kinase that when active will phosphorylate and potentially deactivate RB. So RB is retinoblastoma protein, or otherwise known as tumor suppressor protein. And normally this protein is going to be bound to an inactivating E2F. So let's take a step back here. CDKs. So here's your CDKs. These are things that phosphorylate stuff. When bound to their cyclin counterpart, they're active. And so they have, so here's another CDK down here. When these are active, they have a propensity to phosphorylate things. And in this case, they have this propensity to phosphorylate retinoblastoma protein. So if you look here, this is the red guy here. So retinoblastoma protein, normally, when it's not phosphorylated, when it's just chilling, it is constantly bound to a protein called E2F. So this E2F here is actually a transcription factor. So it's a transcription factor that, when freed, will bind to the DNA within your nucleus and recruit enzymes and proteins associated with the expression of S-phase genes. So a transcription factor, you could say it's kind of like an activator of gene expression. So normally, your tumor suppressor protein is inhibiting this. It's inhibiting your E2F from initiating S-phase proteins. However, when your CDKs, which are activated by their cyclin counterparts, phosphorylate retinoblastoma protein, what happens is your retinoblastoma protein will dislodge from E2F. So then E2F will now has the capacity to float right into the nucleus, bind to all of the regions, the loci within your genome associated with S-phase genes, and prompt their transcription and therefore translation, pretty much bringing you into the S phase. So again, these kinases, your CDKs activated, will pretty much deactivate retinoblastoma protein, and then releasing E2F or activating E2F to do its thing. So what do you think happens when retinoblastoma protein loses its function? So let's just say that there's some kind of mutation that might lower the affinity for retinoblastoma protein to E2F. What do you think would happen if this RB, this retinoblastoma protein, never really had an affinity for E2F? Or let's say you have a mutation where you just have a knockout of that gene entirely. Well, what would happen is this E2F would freely bind to your genome, perpetually expressing genes associated with transitioning from G1 to S phase. And that would eventually lead to G2, to mitotic phase. It would lead to cell replication, and eventually you just get uncontrolled cell growth. So that's what makes this whole checkpoint process, you could say this checkpoint, kind of a prime target for, you could say, cancer prevention. Because essentially, uncontrolled cell growth is cancer. And so one of the more successful targets are actually, it's a CDK4 inhibitor. So if you were to inhibit CDK4, which is that kinase, then what would happen is your retinoblastoma protein wouldn't become phosphorylated, And if it did not become phosphorylated, well, then it would stay bound to E2F. And so you would never transition from G1 to S phase, or at least you wouldn't do so in an uncontrolled fashion. So CDK4 is just one example, but there's quite a few different targets of the CDKs to prevent transition from G1 to S, or even from G2 to mitotic phase. Okay, so speaking about cancer, let's talk about Henrietta Lacks. So she was a patient of the Johns Hopkins Hospital back in the 50s for cervical cancer. So she had cervical cancer, and she actually became famous because her cells were cultured, or they were extracted and cultured, and then studied. They were studied and kept alive. Her cells are actually still alive today. Here's actually an image of it. The magenta is actually a stain of the cytoskeleton, and then the teal is actually a stain of genetic material. What makes her case kind of controversial is that initially her cells were taken and cultured without her consent, and then studied and used to make treatments for cancer. However, herself or her family's or her family was not initially compensated for it. So that's what kind of makes it kind of controversial. Fun fact is that there was a evolutionary biologist named Van Valen who actually referred to her cells as kind of, you could say, a modern emergence of a new species. That's because her cells are considered to be triploid, or 3N. So her cancerous cells are considered to be 3N, and they do not resemble her original genome. Remember, these are cancerous cells growing on their own in a petri dish, in petri dishes. And so they're estimated to contain 76 to 80 total chromosomes per cell, as opposed to the normal 2N 46 chromosomes per cell that our somatic cells would have. So they have a different chromosome number, or chromosome amount, and then in addition to that, 22 to 25 of those chromosomes, actually the chromosomes themselves are arranged in a way that they have different genetic material than their original chromosome number. So because they are, because these cells have the capacity to indefinitely replicate themselves, and because they do not have the same chromosome number or chromosome amount as the mother cells, therefore they are considered to be, a new species. Pretty interesting. All right, so still on the topic of cancer, we have these things called growth factors, which have the capacity to bind to your G1 cells and then trigger the expression of genes that cause the progression through the cell cycle. So from G1 to your S phase. So they'll cause that transition. Cancer cells, fun fact is some cancer cells, depending on the type of cancer, has the capacity to make their own growth factors. And so that's what kind of makes, growth factors another potential target for cancer prevention.